Okay, so far as I know.
That's the telling me, turn on. I'm way ahead of myself. So while we wait, oh man. What the heck?
Holy crap.
It's
Like
two.
secure password.
It's two. It's two passwords that I've just turned into one password. You can do that. So this is for your entertainment. So this is what I'm working on. This is the next generation of video for Bio 1102.
There exist multiple strategies for being large, the most common of which is being multicellular, that is to have a macroscopic body that consists of multiple individual cells. It is also possible for a macroscopic body to consist instead of just a single cell. In this case, in order to have gene products spread throughout the organism, it's necessary to have multiple nuclei throughout that single cytoplasm. This state is known as being senocytic.
It was
There are a number
created
of
off of
organisms that in fact are
figures that I had
senocytic,
for
that is,
this
that
class,
are relatively
obviously.
large and consist of only a single cell filled with multiple nuclei. An example of this are the Glaucharia algae. These algae, again, are macroscopic. They contain a single cytoplasm. The cytoplasm is a large central vacuole that basically is found throughout the algae. And surrounding this vacuole are nuclei as well as chloroplasts. A second example of cenocytic, more or less macroscopic organisms are fungi that lack septa between individual cells. Instead, their body, or thallus, consists of a single cell, that is, a single cytoplasm surrounded by a single plasma membrane, and within that cytoplasm are suspended multiple individual nuclei.
We don't yet have the license for the
Plasmodial
software.
slime molds are also cenocytic. Here we have, again, multiple nuclei found within a single cytoplasm in relatively large macroscopic organisms. You also see the cenocytic state in Plasmodium. Plasmodium is the parasite that causes malaria. During their infection of red blood cells, they go through multiple rounds of mitosis without corresponding cytokinesis. This is then followed by multiple rounds of cytokinesis that converts the relatively large multinucleated cell, the schizon stage, into individual single nucleated cells. So this is one approach to being large. It allows the organism to transport materials throughout its body by using normal intracellular transport mechanisms rather than
We're
relying
just kind of
instead or having to develop
playing around while we wait for
extracellular
people to show up.
transport throughout the body of the organism. And as we've seen, there are a number of organisms that do display the cenocytic strategy towards being relatively large. But nonetheless, the majority
Try to see the
of
sign. Where in the application notes is that?
That's Big Bend National Park.
Oh, it's text. Really?
Yeah, isn't that amazing?
I didn't really know that.
Yeah, it's all volcanic too. It's just
Wow.
totally awesome.
That's really cool.
But that means that the whole point, so it kind of draws you in, doesn't it?
Unless you get car sick. then you-
Well, yeah.
I was thinking that when he first put that up there, I was like, I wonder what happens if you got a student that gets car sick.
Yeah, so I'm gonna have to post these things
They're
doubly.
vomiting.
Yeah, that's right, that's right. I'm gonna post them without the background. But this is all for Bio 1102, see if I can come up with something that's compelling for that. And I've also figured out that for the video, for taking the video, I need to do it with the autofocus turned off. And so it's just on infinite focus, because you can see it kind of changes
Yeah.
back and forth. But I'm learning. I got
I
the
just now
shirt.
realized you were wearing that shirt.
Half price. Yeah. It was great. And it was raining like crazy. And of course, I was camping out.
Of course.
That's what brought the rain, right? It was like, what's it, 70% chance of rain, something like that. And I said, oh, well, that means there's a 30% chance that I'll be dry.
That's reasonable.
And then it came down like they couldn't believe it, and then it just kept coming down. I'd say this is a critical mass. Should we start?
Well, it's time.
Yeah.
It's 4:30.
Okay, yeah. Okay, so just questions, ask me questions. I mean, if nothing else, we've got like 90 questions out of the text. Just, get me started. Get me to do something other than just straight lecture.
Well, I have trouble understanding all the difference between polyphyly and paraphyly and that whole concept.
Thing, yeah.
Yeah.
So, and that's because they're confusing. And part of the reason they're confusing is that if you really start thinking about it, maybe just because I'm an amateur, but if you really start thinking about it, it can be difficult to tell the difference between paraphyletic and polyphyletic. And so that's part of it. But when you're trying to make a phylogeny, what you want to do is make a monophyletic taxon. And you call a monophyletic taxon a clade. And so your goal is to make clades. And if you're failing to make clades, then you're doing something incorrect. And then it's a question of whether you're doing something incorrect on purpose or whether you're inadvertently doing something incorrect. And if you're doing it on purpose, it's usually a paraphyletic. And if you're doing it inadvertently, it's usually a polyphyletic. Although that's not 100% perfect, because it's possible to inadvertently create a paraphyletic taxon, as I can get to. But a clade, the best that we can do in terms of chalk. I'm hopeful that's not the case. It's going to be a little limited. Maybe we... Oh, come on. Oh, well, I guess I'll draw it very lightly. So a clade, you've got your ancestral species, and then you've got various other species, and however you want to draw it. And some of them are still alive, and some of them are not still alive. The ones up here would be still alive. And all of the species, the extant species, and really everything that has descended from some ancestor together make up a clade. And a clade is a monophyletic taxon, and that's a good thing when you succeed in drawing this. Now, it's possible that you have some other group and you've decided not to include that group with this group here, which I would draw a circle around, but I don't want to use up my chalk. And in this case, we call that a paraphyletic taxon. So you've got some group that really should be part of the same clade, but you're excluding that group from the clade. And usually we exclude the group from the clade for a reason, and that reason is that they have evolved in such a way that they really seem to be something different from the clade that they evolved from. And so a classic example of that is you've got the reptiles and you've got the birds. Or another way of saying that is that you include the dinosaurs with the reptiles, but you don't include the bird descendants with the reptiles. And then another great example is humans. We typically don't include the human branches in the same grouping as the great apes. And so you've got the great apes, and there's some taxon, and their description is not as equivalent as it ought to be of the description of our own taxon. So that's a paraphyletic taxon. You've got something that has innovated evolutionarily enough, usually phenotypically, in such a way that it seems to be quite different from the main group, and so you call it something different. But we're all basically reptiles, or we're all amniotes, and we're all tetrapods. But we've got these groups that we say, oh, we're not all modified amphibians, right? But we really are all modified amphibians, and we're all modified amniotes, and we're all modified mammals, and so on and so forth, all meaning all of us humans. And then that brings up the polyphyletic taxon. So with the polyphyletic taxon, what you have is products of convergent evolution that are fooling you. So you've got two groups that are sufficiently similar to one another in terms of looking at them that we say, aha, they must be related. But they only look similar to one another because they've evolved in a very similar environment, and they have come up with similar solutions to the problems in that environment. So they've come to look very similar to one another. But they're actually not closely related at all. So we've got, for example, the toothed whales, especially the porpoises, look very similar to the ichthyosaurs, these lizards that have evolved into this fish-type shape. If you went back to the common ancestor between the ichthyosaurs and the porpoises, what you would not see is an animal that looks like a fish. You would see just an ordinary tetrapod. And that's really what you see when you have a polyphyletic taxon. You see two groups that started out looking different from the way they currently look, and both of them came to look in a similar way, but using completely independent evolutionary paths. And so you say, aha, they're similar, they must be closely related, and you're wrong. And you call that a polyphyletic taxon. But you can get into trouble because sometimes things really do look like they're paraphyletic and polyphyletic at the same time. And so that confuses the issue. But monophyletic, correct. Paraphyletic, incorrect, but for a reason, at least for good reason. Even if it was a mistake, it was still a good mistake. And then polyphyletic, it was simply a mistake, we were fooled, and the root of polyphyles typically is convergent evolution. Working on camera angles right now. My daughter here should be saying, I couldn't see a single thing you wrote on the board because you're between the camera and it. Other questions? That's a good start.
I had some confusion when it was going over expediency and efficiency of utilizing resources, especially when it got into what kind of culturing you were doing. So if it was continuous culture, if it was batch culture. I was kind of confused on the reasoning for each of those.
Okay, so I guess I have to do my best to try to do this without chalk, because I might actually need chalk for real at some point. Okay, so an efficient organism is utilizing its resources more efficiently, and so it is essentially making more progeny for a given amount of resources utilized. And the expedient organism is utilizing resources inefficiently, but it's doing so for a reason. Now, it's possible to utilize resources inefficiently just because you're not good at utilizing resources. But it's also possible to take that utilization of resources that's inefficient and use it towards essentially faster population growth. So you're not necessarily growing your population to what could be the maximal population size based upon what resources are available, but you're getting to whatever your maximum population size is faster. And a good example of this is fermentation versus cellular respiration. So with cellular respiration, you're getting, 32, 34, 36 ATPs per glucose if you calculate it in terms of glucose. And with fermentation, you're getting, maybe 2 ATPs per glucose. And so you're clearly much less efficient when you're fermenting. But apparently fermenters can grow faster. At least they can ramp up in their growth. They can get going and grow their populations faster than an organism that has to create all of the mechanisms, all of the, what is the right word? All of the mechanics necessary. What was that?
Pathways?
Yeah, pathways works. That wasn't what I was trying to think of. Pathways fine. All the pathways necessary to go through cellular respiration. And it's very complex stuff. It takes some serious effort to put all of that together, and it slows down the rate at which the organism can grow. If the organism has enough resources available to it, that it can get away with just making 2 ATP per glucose, then it has the potential to grow faster. And so those organisms can be seen as expedient. They are very inefficiently using their carbon and energy source, but they are doing so in a way that allows them to be more effective, at least in terms of rate of population growth. Of course, just as an aside, fermenters also can take advantage of, they can grow when there's no oxygen around, whereas the obligate cellular respirers can't grow. But there are these facultative organisms, and the question is, why not just be facultative? And the answer is that it gets in the way of your expediency, your ability to grow fast. So you've got these 2 different things, and we can think of the more efficient organism as potentially being a greater cooperator than the less efficient organism. And a way of thinking about that is you're an individual who is in a group, and within this group, if you use up resources too quickly, they're no longer available to the group. And if the group is in fact your own progeny that you're taking resources away from in terms of inclusive fitness, I mean, that's taking away from your own fitness. So it's cooperative in the sense that you are not just burning through these resources and therefore making them unavailable to others that are your conspecifics or even your own progeny. And it's also cooperative because you're not just burning through all these resources in order to simply grow faster than everybody else. So the expedient individual, that's more of a non-cooperative, more of a defection strategy, because they are trying to utilize the resources in the environment in order to gain an advantage over others, and they're doing so in a way that hurts others in 2 different ways. One of them is they're simply using up the resources, and the other is that they are displaying faster population growth, and so taking over the population. So when we talk about the evolution of virulence, it gets a little complicated. But an important component of this is that there can be individuals within a body that if they can replicate faster, then they can become a larger fraction of the total population. And as a consequence, they can have an evolutionary advantage, because if you're sampling the population in order to transmit from one individual to another, if you're sampling the population just in terms of what's inside of a body, this is your transmission step. If your genotype makes up a greater fraction of the individuals in the body that are being sampled, then just on that basis alone, you have a greater likelihood of being transferred. Now, the problem with that is that more rapid population growth, or population growth to a greater extent, it gets a little complicated thinking about it, can lead to greater harm to the body that you're growing within. And that can result in the opportunities for transmission to be lesser. And a good example of that is if you need a living body in order to be transmitted to the next individual. If you kill the host, then you can't be transmitted. So there is this conflict going on. Whereas within the body, you're going to have selection for organisms that can grow faster and faster, perhaps to higher levels, perhaps to causing greater damage to the body. But eventually, the amount of damage caused to the body will result in a reduction in the ability of the parasite population to be transferred effectively to the next individual. And you can deal with that, or a parasite population can deal with that, if it is a clonal population and isn't spending huge amounts of time inside of the body. That way it starts out as just a single genotype, and that genotype has some level of efficiency versus expediency associated with it, and some level of virulence associated with it, and therefore some level of impact on the host in terms of damage, and therefore some way of controlling its ability to be transmitted to another host. No matter your level of virulence, no matter how you're transmitted, there can be a point where you're damaging the host so much that it's going to start harming your ability to be transmitted. And a good example of that is that even if you are an organism that kills your host in order to be transmitted, if you still need to replicate inside that host prior to it dying, it's always possible to be so damaging to the host that you kill it so soon that you don't produce enough progeny at the point of transmission. So there's always the possibility of harming the host so much that it actually harms you. Now, if you are a population that starts out with multiple individuals, then you have the potential for, this is their parasite individuals, you have the potential for there being multiple genotypes inside the host from the start. And therefore, they can have different levels of expediency and different levels of virulence associated with individual populations. They can duke it out inside the host, and the individuals that can grow their populations faster will make up a larger fraction of the total population of the host, but that faster population growth can result in greater damage to the host and therefore affect the ability of the host to, the parasite to be transmitted to new hosts. So if you have a population of parasites that starts with a single individual, they have a single virulence associated with them, a single genotype associated with them, and then they can experience between host selection. So rather than having this arms race or this competition going on inside the host, they simply produce a certain level of disease in the host, and then other parasite populations that they're in competition with of the same parasite type, infecting other hosts, will have a different level of virulence at a different rate at which they can be transmitted. And it'll be that population that is more effective in being transmitted that will end up dominating the population as a whole. So there you've got, on the one hand, between host selection, between population selection, and that's for faster transmission rates or more effective transmission rates, and you have within host selection, and that's for faster population growth rates. And you can control this by starting a population as a clonal population, so a bottlenecking step before you initiate that. And you can also control this by not infecting forever so that you don't eventually accumulate these mutations that are detrimental. And you can, or that give rise, I shouldn't say detrimental, that give rise to faster population growth, that give rise to cheating or expediency. And you can also control that by not having a high mutation rate. And that way you don't have a high rate of mutation to expediency. And that way you can control better what virulence that the population within a host is going to be able to display. And if that virulence is virulence that's close to the optimal, that gives rise to the better transmission rate, then that will be the population that infects more hosts than other such competing populations. And that will be the level of virulence that gets selected. And then that whole idea that you have this competition going on between expediency and between economy, and this whole idea that you have these mechanisms that you can employ in order to increase the amount of economy that you're displaying, or the amount of cooperation that you're displaying. They tend to be consistent going from system to system to system, whether it's multicellularity, where the individual body cells, the normal body cells, they're in a sense displaying an economy. It is perhaps not strictly economy, it is however a cooperation where they are not displaying excessive rates of population growth. They are not ideally mutating, or if they are mutating, they're not being able to easily turn into cheaters because they've got all these internal within the cell control mechanisms. And these cells can therefore increase essentially the rate at which they're transmitted to the next individual, which is for them the reproduction of this multicellular organism. And so if they can start out as a clone, a single cell zygote, and if they can go through not too many rounds of replication, and if they can have relatively low mutation rates, then they can minimize the number of genotypes within the bodies that they're creating that are cheater genotypes, and therefore have a greater potential to reproduce. And they also can have immune systems which not only can block cheater genotypes within the body from getting out of hand, which we would call things like cancers or tumors, but the immune system also has the means of blocking what I call cheaters from without, from getting inside of a body. These would be parasites, pathogens, and replicating within the body and being able to essentially increase their own fitness at the expense of the fitness of the normal body cells, and therefore potentially reduce the ability of the body to reproduce. So the trouble with expediency versus economy is that when we go to these other systems, it isn't as clean a transfer of the idea as could possibly be. But they're an example of cooperation versus defection, and this idea of having the ability to cooperate and for cooperation to evolve is a theme that runs through all 5 of the chapters, even the Genome chapter, where you have genes that, in a sense, cooperate with one another by making up pathways inside of bacteria that get grouped together, linked together, so that they have some potential to, in fact, be transferred via horizontal gene transfer mechanisms to other such bacteria. So it's this whole idea of individuals that are going to be cooperating, come together or stay together in this group, and they are helpful to each other and they don't cheat on each other, and ultimately, ideally, this is going to lead to some greater potential for them to transmit their kind into the next generation. I'll try not to be quite that long-winded on all answers to questions. But that's a really important one because that hits the nail on the head of just about everything that the second part of the course is all about. Did I successfully answer the question? Okay. Does anybody have a follow-up question to that one? Or any concerns about economy versus expediency? Or anything?
Back to chapter 9.
Yes.
I did another thing I troubled with was the especially orthologous replacement and then orthologs, which to me seem to have opposite definitions. I don't know if that's on purpose. Okay, but that wasn't going well with me.
Yeah, so the orthologs are simply genes that are in 2 different lineages that are descended from the same gene at the same locus. So, I don't know, if you had 2 beta-galactosidase genes that were in the same position in, say, Shigella versus E. coli. hesitate now that I say that because I don't recall whether Shigella actually is a lactose fermenter, which is probably pathetic on my part. But you've got these genes that basically are identical by descent. They're the same genes. They started out in the same place and they stayed in the same place over the course of the evolution of the genomes that they're in, and those are orthologs. Now, orthologous replacement, you're right, you're coming in horizontal gene transfer and you are swapping out the existing gene and replacing it with a new gene. Now, it's got to be, if the gene is necessary for the fitness of the organism, important, it's got to be a gene that has a similar function to whatever it's replacing. And typically it does, because the genes that are going to come in and replace by homologous recombination an existing gene, the only way they're going to do that is by having a relatively high amount of homology, similarity, in terms of the nucleotide sequence as the gene they're replacing. So homologous replacement, it's something you see with homologous recombination, or sorry, orthologous replacement is something that you see with homologous recombination. And you can think of it as being less of a contradiction because you are, after orthologous replacement, you're left with a gene that has a similar function and a similar sequence, and probably was orthologous from in terms of whatever organism it came from, in the place of the original ortholog. And then paralogs, you have a gene duplication event, and then you've got two different genes that were once the same gene that are evolving independently from one another. And paralogs also are potentially subject to orthologous replacement because they too are orthologs. I mean, in terms of just, you've got two genes and they're both off there evolving, but each one of them is orthologously related to the equivalent gene in the equivalent place in another lineage that broke off from the first lineage after the duplication event occurred. And so you can certainly have homologous replacement coming in and replacing one of the paralogs with something via horizontal gene transfer. Where it gets particularly complicated is that you can have gene duplication events where ultimately one of the duplicates is lost. And this can lead to, when you're comparing two lineages, it can lead to strange things going on in terms of whatever tree you're making, whatever phylogeny you're creating based upon these genes. Because you've got two independently evolving genes, and if this lineage loses one of the genes and the other lineage loses the other gene, you're losing the information that once upon a time they in fact were the same gene in the same lineage, became two genes, and so they went through their independent evolution within the same lineage without horizontal gene transfer, and then you're just losing one of them, one of the copies in one of the lineages, and the other copy in the other lineage. And so it looks like the genes really have been evolving separately from one another for a long time. And that evolution, that the degree of distinction between those two genes would be much greater than it would be if you just had the two lineages and they each lost the same copy. Because therefore there's less independent evolution that has gone on. But when you've got these loss of paralogs, it can look like horizontal gene transfer events, because another way you can get that kind of effect is if you have a gene that has been replaced by some other gene that's similar to it but has been evolving in a different lineage. And you can get these independently evolving lineages of genes. these gene phylogenies that are different from what the organismal phylogeny looks like. But the idea of paralogs is really the old way that people thought about how you can get these independent, these gene phylogenies that are different from the phylogenies of the organism as a whole. And that was before people had an appreciation for how pervasive horizontal gene transfer is. Now, typically, when we see what appear to be independently or somewhat independently evolving genes within a larger genome, we usually think that, aha, those genes must have been acquired by horizontal gene transfer. So another thing is when you get insertion events. In an insertion event, what you have is a new gene coming in, and it does not end up in necessarily the same location as it was in whatever lineage it came from. And that's when Lawrence and Hendrickson talk about the zone of paralogy. It's confusing because they use the word paralogy, but what they're talking about are the reasonably high likelihood, at least as far as they argue, that a gene that comes in from another location and inserts into a genome has much less selection against it than a gene that orthologously replaces something that's already existing. And this is because if you have orthologous replacement, you have to, if natural selection is driving the whole thing, you have to not only be able to do whatever function the original gene did, but you have to, in a sense, do it better if you're going to have it selected for. But if you have an insertion event, then you can have this new gene coming along, and although it's adding a little bit of DNA to the organism, and ideally it hasn't inserted into a place that disrupts existing functions, what you have is a gene that may be able to supply a new function to the organism, and it doesn't have to simultaneously supply some essential function that already existed in the organism. So as you, in that zone of paralogy figure, so you've got homologous recombination. As phylogenetic distance increases, the likelihood of the success of that gene transfer declines, because homology declines, because likelihood of transfer declines, and also because the potential for that new gene to take over the function of the old gene effectively declines. But at the same time, as you get increasing distance, you have a lower amount of homology, so if you're going to acquire the gene, it's likely to be acquired by illegitimate recombination. And so you've got this potential bump, who knows where exactly it lies in terms of phylogenetic distance, but it's able to come in and supply a new function. But then they're arguing that if the phylogenetic distance are too extreme, then you reach a point where there's just no way this new gene could possibly supply some new function because the organism it came from is just too different from the organism that it's going into. And so the likelihood of successful transfer declines simply because the potential utility of this newly acquired DNA declines.
So you had a question that it's when one or two horizontal gene transfer explanations for the lack of correspondence between gene-based phylogeny and organismal phylogeny. So is an insertion event?
Yes, so right, insertion event is the second one. Yeah, you either have homologous recombination or you have illegitimate recombination. Now, with an insertion event, you're going to have a new gene that simply is otherwise not present within the original genome, and therefore when you're putting together the phylogeny, it's simply going to suddenly appear. But if you have that insertion event, and now you have what appears to be paralogs without knowledge that we had a horizontal gene transfer event, and then you had loss of, in other words, two genes that are similar to one another to some degree, and then you have loss of the original gene, suddenly you can be making these phylogenies and you go to this gene and you say, aha, this is the equivalent gene over here, and it seems to have a completely independent evolutionary history. It could be because it was a product of illegitimate recombination insertion event followed by loss of the original gene, and therefore it really does have a completely different. But that's, yeah, so that's how you end up with gene phylogenies differing from organismal phylogenies by horizontal gene transfer. This is good. Keep it up. Go for it. Yeah.
In chapter 9 also, there's a page where you talk about some things. And
That's okay.
it's
hard to talk, I mean, off the top of one's head in a way that the
So,
words
well,
all
you
the time.
have this table, and under hitchhiking, you describe, like, it overlaps with paralogous genes, which are the products of gene duplication events.
This is the one with the, I mean, it's a table where I've got multiple, here, let me see
Yeah,
what
I
you.
think so.
Oh, okay, yes, it's a table, it's just a table, okay, go for it, go for
Okay,
it.
and then you say, well, because it's under hitchhiking, it says these genes do not contribute to the fitness of the carriers, in the sense that their deletion will have no or very little negative fitness consequences, but there are gene duplication events that allow for like sequence space exploration where those can still be, like they can actually contribute to
Yes,
the fitness of.
Yes.
Okay.
yeah. So usually you think about if impact on fitness is either being positive or negative. And what I was trying to do is kind of distance myself from that idea to describe these hitchhiking things, because you can have an allele that hitchhikes that is no more beneficial than, I guess it's neutral in terms of its fitness compared to other alleles within the same population. But you could also have an allele that simply has been acquired, and really it doesn't have any impact on how this organism functions. It might not even be a gene. And it can be carried along by periodic selection, by linkage. It can hitchhike along with some beneficial alleles in the same organism.
But, so what I'm saying is, even if you have a paralog or two paralogs, and if the other one is evolving separately, and it can also provide a benefit.
Yes.
It doesn't have to just hitchhike.
Yes, yeah. So those are the four things. So you've got the core genes, you've got the genes that are kind of core genes, but they are more easily orthologously replaced. So the gene remains there, but it can have an independent evolutionary history from the rest of the genome. Then you've got genes that can be acquired by horizontal gene transfer, or as you're saying, acquired by duplication. But usually you think of it as horizontal gene transfer, the way it's coming in, because that's our new, over the past couple of decades, understanding of what goes on. And those new genes that come in, they can provide benefits. So like you're saying, if you have a duplication event, you could have both of them providing benefits. And that's really the third category that I list in those four categories. But then you can have stuff that comes in that doesn't provide a benefit and also doesn't provide a cost other than some small metabolic cost. And they can be selected for simply because they're linked to other things that are being selected for periodic selection. And then you can have them increase within the population simply because of that. So yeah, in no way am I trying to say, in no way am I trying to exclude the idea that you can have beneficial alleles coming in by horizontal gene transfer, beneficial alleles being created by gene duplication events, because you can. But then there's also this fourth category of stuff that can come in. And then really there's the fifth category of stuff that can come in and actually is detrimental to and those are lost. And so you never really see them when you're sequencing organisms. And it's usually by sequencing organisms that we infer that a horizontal gene transfer or a duplication event has occurred. And so if you never see it at all, it's not going to be one of the types of genes that is going to be found within these lineages. So yes, definitely they can be beneficial. Go for it. That's fine.
You mentioned that defective interfering particles are more virulent than wild type.
Yes.
So can you explain that?
Well, what they are virulent against is not necessarily or so much the host cell that they're infecting. So the defective interfering viruses are a type of virus. So they have to replicate within some kind of cell. So it's not the host cell that they're virulent against, it is the virally infected host cell that they're virulent against. So you can have a cell that's infected with a wild-type virus, and then you can have a cell that is infected with a wild-type virus and another wild-type virus, with the hands being the different viruses. And then you can have a cell that's infected with the wild-type virus and the DI particle. So the original infection, when there's a co-infection with another wild-type virus, I mean, there's some sharing of resources, but it's not the end of the world. With the DI particle, there's actually an excessive sharing of resources. So there's a damage that is being done to the wild-type infection that simply is not seen in the absence of a parasite or a co-infected virus, or as much with a wild-type virus. So when I say that they're an example of virulence, it's not virulence, an increased level or higher level of virulence against the uninfected or otherwise uninfected cell. It's an increased level of virulence against the virally infected cell. And that's what DI particles basically are. They're parasites of virally infected cells. And there are other kinds of parasites of virally infected cells besides DI particles. There's a number of different types of these things. And in order for this to occur, for order for these parasites of virally infected cells to even evolve, you have to have a sufficient frequency of virally infected cells out there to support the parasites of the virally infected cells. So it actually plays into the ideas of evolution of virulence even more. They have a cost, an antagonistic pleiotropy of displaying this higher level of virulence because they can't replicate without the other virus around, and they can only pull off this higher level of virulence if the opportunities for infecting virally infected cells are sufficiently high. I moved from there to here on the assumption that I was off camera or standing right there. That actually is past. That's chapter 12. That question. In a way, you can also think of the DI particles as being more expedient because they are replicating faster or more effectively because they are investing less into their the particle that they're actually building. The particle ultimately is not as effective as the more efficient wild type is. And you can also think of it in terms of the wild type being more cooperative and the DI particle being more of a defector. Certainly, not just more of a defector, a defector person. So again, all of these ideas in all 5 of the chapters, I mean, there's this core of ideas that we consider over and over and over again, and they play on expedience versus efficiency, cooperation versus defection, and the like. Yes.
Pretty simple question
Sure.
about the tragedy of the commons. So, one of them you asked what the tragedy was specifically, and I was wondering if it was the destruction of the commons or the temptation to destroy the commons. What
No, it's the actual
The actual
destruction.
destruction,
Yeah,
okay.
yeah.
Okay, that's-
Yeah, that's the difference between, well, I guess it's actually, I was gonna say temptation to defect, but actually temptation to defect describes an actual defection. But it is, I mean, it's what you get if you defect. You get an environment that is more degraded, or a public good that is less effective, or a host that is more sick. I mean, these things, again, they all go together. And the tragedy is this reduction in some other measure of the functionality of these organisms than just whatever it is that is being accomplished by doing the defection.
I'm thinking about the expediency of the defective and nuclear particles, as you said, but can't they only be because they rely on the virus, the wild-type virus, for like capsid and whatnot? Can't they only be as expedient as the wild-type virus? Because that's kind of their limiting factor.
Well, there's a certain amount of resources. There's a public good inside of the cell, and that public good is actually being supplied by both the DI particle and the wild-type particle, because these are the gene products that are produced by these viruses. But more of the public good is being produced by the wild-type particle than by the DI particle. And so the DI particle is taking more than its fair share, and it's also not contributing as much to the total amount that's there. And I mean, I guess you can get into semantics and say, is that really a lack of efficiency? And I don't know, but it's a more expedient approach because they are taking shortcuts. The shortcuts are hurting them if they can't manage to find another cell that's virally infected, so they can hurt them in the future. And they're doing it for short-term gain. Short-term gain is in terms of an ability to replicate or package or just increase the number of viral particles faster than the virus that they're co-infecting with. So they're definitely defectors, and describing them as expedient seems to work on an English level, of basis. And they are doing so at a cost. Now, if they didn't have that cost, if they could go in and infect these cells and gain, but there was no cost associated with that gain, then you would have the expectation that the DI particle lifestyle, or whatever we would call it, because it wouldn't be defective, would in fact become dominant within this population of viruses. So I mean, it's always possible to be more expedient. The only thing that's going to select against expedience is some kind of antagonistic pleiotropy, some trade-off that's associated with that expedience. And as long as the trade-off doesn't exist, you're going to have an expectation that ever greater levels of expedience will evolve until you reach a point where, in fact, the trade-off does become imposed upon them. And again, this is just a consistent theme. If it's growing faster, if it is growing less efficiently, if there's no cost to that growing faster, if there's no cost to whatever gains it's getting, then there's going to be no selection against it. There's only going to be selection for whatever that is. So you're going to have the population evolve to the point where you start having the costs actually impinge upon further evolution of that character along the same pathway. And that will be the optimal level of, say, virulence that's being displayed. And if you have less virulence than that, it might, in fact, be associated with lower transmission rates, and higher virulence associated with lower transmission rates. But within the population infecting a single host or within a single cell, you're always going to have selection for something that is more effective at replicating, simply because they'll come to dominate the population. Yeah, I had a feeling, I had the sense that, yeah.
I wasn't really sure of the answer to this question. It says the most noticeable differences when comparing genomic sequences of different bacterial strains are what?
Well, our insertion events. And I think it actually says in like the figure legend above that, that I think it uses the same words. Just as a clue as to where these questions come from. I mean, they basically, I take the section and I go through the section and I place questions at the bottom that clearly come out of that section. And the goal really is to motivate you to read through the section and then just to think about it a little bit. But yeah, I believe it actually uses the same words above it, and I think it's in the figure legend above it. I'm not a genius that memorizes everything. I looked at that a couple hours ago.
The question after that is,
Yeah, I have no idea.
with greater phylogenetic distance between donor and recipient lineages, what becomes more apparent in terms of orthologous gene replacement? So that was just going off the picture of the likelihood of retention.
So, and there's a question that comes immediately after that that's worded almost the same way, I believe.
Yeah.
What becomes more likely or something like that?
Yeah.
Again, I don't have these memorized. I just looked at that earlier today. I went through the questions and I said, oh yeah, okay, that comes from there. So that's the question that comes after that is, and I can't remember what the question is, but it's explicitly what it is. But that's an easy. Sure.
With greater phylogenetic distance between donor and recipient lineages, in what way is the likelihood of orthologous replacement?
And so the answer is it declines. It has a lower likelihood of greater. So that is the easier of those two questions to answer. And I agree that the one above it is not the most straightforward one in the world. So read it to me again, so I can remember what the answer is.
With greater phylogenetic distance between donor and recipient lineages.
Oh, what becomes more apparent?
Yeah.
And so I believe that the answer to that is simply the orthologous replacement becomes more apparent. That if you've got two lineages where the genes that are being orthologously replaced are too similar to one another in terms of nucleotide sequence, you simply can't tell that there has been a replacement event. And the extreme of that, it's always good to think about things in extremes because you can kind of base some of your logic on this. But the extreme is that you have an orthologous replacement event where you have horizontal gene transfer, but you have replaced a gene with an exact copy of the same gene. So horizontal gene transfer has occurred, but you can't possibly know that it has occurred based upon sequencing in the future. But if you've got one nucleotide difference, you're probably not going to be able to say, oh, well, that's horizontal gene transfer event. That's horizontal. You can't say that's orthologous replacement. You probably say, oh, a mutation occurred. But as phylogenetic distance increases, the number of nucleotide differences between the gene that was in there originally and the gene that is replacing it, the number of nucleotide differences increases. And therefore, your ability to tell that an orthologous replacement event has occurred also increases.
So the amount of orthologous replacement isn't actually increasing like
It actually is decreasing.